All right. All right, our class, we're going to start up class again. And I have your second exam. I'll hand that back at the end of class today. And talk about that some. I made an answer key video to try to talk through the exam, help you understand that. There are a couple of reasons you want to watch that answer key video. One is to follow up on this exam and make sure you understand this material because the final exam is cumulative. So we're adding stuff all along as we go. And then also because the third exam and the fourth exam, the final, are going to be the same format. They're going to be the same style of questions. And I feel that some students start to recognize the style of questions that I would ask on exams and have increased their scores. Some have, like that. And I don't think that's surprising, it's kind of inevitable that whoever the instructor is has a certain way of understanding the material and then wanting to see if you understand it, they would ask questions a certain way, right? And so we've seen that, and I think going over this exam and just processing it and seeing, the answers helps you understand how to get ready for the third exam, even though it's going to be, a new material like that. So I think it's really good to spend time on it and then follow up on it. When it comes time to, adding the points you got right, I could easily make a math addition error like that, and I apologize that, but, when we have like, oh, it's a half point here and half point there, just going through that, I can make a mistake or just in the, your answer, right? I'm like, I think I understand what you're saying, and I don't think that's a full credit, like that. And we want to talk about that, I think there's a reluctance from students, kind of they feel like, oh, it looks like I'm grubbing for a grade or something like that, and it's not like that at all. You're, trying to learn, trying to understand this, and I'm trying to help. And so we want to follow up on that. And then when you hand out get your exam back today, you'll see you have a circled score on it. That's the number of points you got on the exam. And I never base my grading on the percentage, 90, 80, 70, 60 cutoff. I also never so-called curve a grade where I multiply that score times something. That's your score. And what you have seen is that I'll attach a grade distribution, anonymous grade distribution, to the answer key video announcement, and it will show asterisks for what students got for as scores. And if you got 65, there'll be an asterisk. Maybe there's two. Maybe you and some other student both got 65s. But that's the circled score on your exam, and right next to it is your letter grade. So you have a good understanding of your letter grade from the first exam. You'll have a good understanding from your letter grade from your second exam, and that's is your indication of how well you're doing in our course. You should be comfortable with that. You should recognize that we have 2 more exams, and I think on the syllabus the count of the exams goes sort of like the first is like 20%, the second 25, third 30, and the fourth 35. So that's because I want to super emphasize that I'm really impressed if you can figure it out. Like I said, get kind of familiar with what the expectations are and increase your score, you'll increase your grade. In addition to that, I look back, I have described on the syllabus just a formula that initially Excel just calculates the final grade. Boom, there it is. But I don't just submit that. I look back at everybody's scores and grades, and I look at it and think about it, and it's true that when I grade exams, I grade them anonymously, one question at a time, but when I'm done, I look back and I see how each student is doing to try to get a handle on that and awareness of that, so that, at the end of the course, when I think back, I think of you, I think, yep, I see, like that, and I try to give you as good a grade as I can. And I am impressed, if you can hang in there and try to increase your grade like that, yep. So it's a tough course, and I'm trying to, teach it in a way that, helps, learn the material, learn about how you learn, and then, you know, try to give you a good grade, as good a grade as I can. So that's where we're at in that. So again, we'll have the recording link today, and we'll also have another breakthrough announcement with the ASCII video and grade distribution like that, okay? And want to follow up on that, okay? So the deadline is passed for these internships, but if you're going to be here next school year, you can still apply. We have some good applicants, but we have a good chunk of funding. It's just the most impressive internship program that I've ever seen. So I really encourage you to, you know, consider it. And you and I can talk about it because I'm the contact person. And so if, you know, got back from spring break and you're like, oh yeah, what's, you know, like that, we can still talk about it and still get an application in. It takes the scientists there a while to go through it. I don't do any of the selection. It's those individual scientists there, pictured there, and others that do it, and it takes them a while. So we can still participate in that. Okay, so what should we do this week? So ASCII video and follow up on this exam, follow up with questions. You can ask those questions in class, you know, what the heck is that, you know, or you can ask me, you know, we can, you know, save a time, okay? Then we're going to do chapter 10, the nucleus. So we're going to talk about that chapter. It's in section 3 of our book, and I described last week that section 3 I would call this, it's called like cell biology. So the nucleus. So let's take a look at the nucleus and see what is that organelle, what's it up to, how does it do its job. And so cell biologists really want to look at intact cells or intact parts of cells, the ribosome, how does it get its big job done? Inside the nucleus, the nucleolus, where ribosomes get assembled. The mitochondria, you know, like that. They want to look at intact cells or intact organelles. And they kind of would fight with a biochemist, and they'd say, wow, you can't just purify these molecules. You got to look at the intact structure in these cases to really understand what is the function of this and how it fits into the story of life, like that. And so for our third exam, I think we'll cover 3 chapters from this section. So they'll all have a similar kind of style to them, the nucleus. And then I think in chapter 11, that's about protein synthesis. And that's also about the cellular, you know, I just mentioned ribosomes, the cellular process of protein synthesis and getting the proteins to their proper destinations. And then the third chapter we'll cover from here is the cell membrane, the plasma membrane. And so what we'll do is we'll use the same spirit of going through this. We have lots of good examples that we can plug into it. These chapters, you'll see, you know, oh, cell membrane, oh, well, we know some receptors on the cell membrane. So let's kind of emphasize that, you know, that's a place for ligand binding and receptor function and stuff like that. And we know, oh, scramblase is in that inner leaflet. We also know that some of the components in the signal transduction pathway are anchored to the cell membrane like that. So we have kind of a good handle on some of these, and we'll kind of emphasize that. As part of chapter 10, we're going to have a drawing assignment. It's going to be due a week from Thursday, and it's called RNA processing. And RNA processing is not dealt with in chapter 10. RNA processing is actually dealt with back in chapter 6, and we're never going to read chapter 6, and you don't have to look at it. So this is really an assignment that just kind of stands alone, but it's all really important stuff for us. And so there's some drawings in there to get those done, and like we've done before, send me pics of that. And I think we'll just have we'll have 1 more of those assignments, maybe 2 more for the rest of the semester. Because you know, after spring break, we start going like toward the end. So then we have exam 3, and it's coming up, you know, in like slightly less than a month. So we gotta plow through this stuff, but it's all good stuff. It's somewhat different that we've been dealing with, so it's a way to add, you know, whoa, I can put more knowledge over here and over here, like that. So that's where we're at, okay? And so this just looks at, I showed this before, you know, the structure of our textbook, and we just took a target right for our course right to that section there about the story of molecular mechanisms of cancer. And we'll keep that up, but here we're expanding it to cell biology and talking about cell biology as we roll forward with that, okay? So here's a picture of a cell, and it's got the nucleus on it, it's got protein synthesis on it, it's got the cell membrane on it. So see, cell biology is cool because we can turn our attention, our minds can go to the nucleus or to the ribosome in the cytoplasm or to the cell membrane. And so it's another way of storing your knowledge. And it's a big picture, right? You can make it big like that. You can imagine, you know, the nucleus is as big as your apartment, like that, you know, or the town, yeah, like that. And that's a good thing. And what's depicted on here is something from intro bio. I'll just, I think on the next slide, here's a figure from one of the chapters in Campbell intro bio textbook. That's the textbook that we use here at USM. If you're a transfer student, you could have also used that. Every intro bio textbook shows this, and it's the pathway of gene expression. And we've really focused on transcription and the regulation of transcription. That's the first step in gene expression. And in talking about the nucleus, for our homework assignment, we're going to bring in the next steps. And the next steps take place in the nucleus, and they're called RNA processing. So we get this transcribed, we get the RNA that we've learned about, and then before it leaves the nucleus, it gets modified, it gets altered. So that's another step in gene expression, and it comes after transcription. So we should put some terms on this. And it goes like this. So you can say transcription, right? And what we're interested in is the regulation of transcription. It's not just the kind of robotic process of RNA polymerase binding and transcribing the DNA into RNA. How's that switch on and off? Like that, okay? So then this is, well, let's just say this is transcriptional regulation, and then let's do this, where that's transcriptional regulation there, that's the gene, and then everything after that is called, generally, is a general term, post-transcriptional regulation. So there's that first step, and in general, that's the most important step. And then, for all these next steps, there are specific examples where the gene expression is regulated. So post-transcriptional regulation of gene expression is significant, and it occur all along that pathway. So transcriptional, we're done. We got the RNA. Then the next thing is this RNA processing, and that's what the assignment is. It's happening inside the nucleus, and what happens, like that, okay? And then it gets, the RNA gets up to the cytoplasm, and if it's a mRNA, a messenger RNA that has a coding region that's coded for a protein, then we have translation. And there can be translational regulation. So we have regulation that's called RNA processing. That's post-transcriptional, and that is in the nucleus. Then in the cytoplasm, we have regulation of translation. Then the protein is done, and we have what's called post-translational regulation. And all of those fit into steps that can be regulated, right? And we've seen examples of several of those already. MYC has a lot of regulation all along its pathway, right? So it's really hard to get MYC transcribed. Then the RNA is unstable, just gets broken down right away. Then ribosomes do not want to translate a MYC. Then the MYC protein, really unstable. So there's regulation that can occur in specific cases all along the pathway of gene expression. And we want to learn about that as we're learning about the nucleus and what happens in the nucleus, the cytoplasm, what happens in the cytoplasm, and the cell membrane. What about the cell membrane? And our special focus as molecular biologists is we're really interested in the gene, okay? Like that. So how does this fit together? All fit together from a molecular biologist perspective, and it goes like this. So you've seen an evolutionary tree, right? It's got mammals, humans, stuff like that on it, right? So that's zoomed way out, right? We're zoomed way out when we're looking at evolutionary tree. And if we zoom way in, you and I are at the tip. And the evolutionary tree, when zoomed in, is all the individual organisms that live. And so when we zoom in and we just look at humans, we could talk about the evolution of a human. And we could focus on, say, evolution of the human eye. And people talk about that. How on earth did such a complicated structure ever evolve? And the answer is, well, if you look and you zoom on the evolutionary tree to the level of the individuals, you see for all multicellular plants and animals, the organisms are like the individual, like pearls on a pearl necklace. So here's you, there's your parents, there's your grandparents, great-grandparents, and it's like that. So for every multicellular organism, there are generations. And with each generation, we make new parts. You were all new, right? Right? New eyes. They just weren't here. Now, we can talk about the evolution of human eyes, and people usually say, oh, the human eye has been around as long as humans have, a couple million years, like that. That's true. But that's because our reproduction is not synchronized. If the reproduction was synchronized, it would be human eyes are here, they're not, they're here, they're not. And that's the truth of the story of multicellular life on Earth. Is it's not here and it's here. And in between, we have cellular life. All life comes from pre-existing life. Yeah, cellular life. And on our planet, every cell uses DNA as the primary heritable molecule, and every cell has more DNA than it's using at any one time. So this regulation of gene expression is the big show, the biggest show of life on Earth, because you're going to make a new eye, and it's mostly based on somehow the information stored in the DNA, like that. And DNA storage information is like a computer. You got all of the storage, all of the files on a computer. You only open the ones you need at a time. That's how DNA works too. So to make a human eye, body, every plant and animal on our planet, we're going to use the information. We got it, genomic equivalence. It seems like for multicellular organisms, just storage, like the memory storage on a computer, seems like memory storage is cheap now for computers and also for life on Earth. Multicellular life on Earth seems to have no problem having a big genome with lots and lots of genes like that. So we're going to use it over time and space like that. So this regulatory region becomes really significant for the story of life on Earth like that, okay? And so if you watch what happens to make each generation anew, you see that it's not really the human eye that's evolving. It's the process of creating or developing, forming eyes and bodies new every generation. And somehow the DNA has all that information in it, and we can compare it, we can study it, it's physical like that. So that's molecular biology talk to say, let's just start looking at genes like that and see what's going on with the genes. And that's what we've been doing. And we started out with, myogenin, see how myogenin gets activated to make muscles. And the story is true for every gene. We got the product, the computer file that you want to open and look at, your resume, right? It's a file, could be a gene, the transcription unit, a unit of information output, and then attached to it, physically attached to it is the cis regulatory region that dictates where, when, and how much. And so that's the big story. Where, when, and how much of this are we going to use to put together, eyes and bodies and plants and stuff like that, okay? And so then you say, okay, cis regulatory region, that's really important. Then you see it's not changing. during our lifetimes, yeah, I could get some DNA methylation, great, you know, like that, but in general, it's not changing. We have the same cis-regulatory region for our insulin gene in our pancreas and our brain, basically, you know, like that. It's not changing. What is the switching dynamic story to the big show? And the answer is transcription factors. And so we started with our course just to show that, you know, that talking about making muscles, myogenin is essential. But part of the myogenin story is that MyoD has to activate it. And so then we have this idea of transmutation, mutations somewhere else that are essential for the activation. So we focus a lot on transcription factors. We keep talking about it. And then early in our course we said, now look at this. Here's a transcription factor. It's the estrogen receptor. And all transcription factors are proteins. They must have their own genes. They must act in trans on their target genes. And the transcription factor is also something that gets inherited as a gene, and it's got to be expressed. And then with the estrogen receptor, we saw, oh my goodness, it's not just the expression of the transcription factor that's the switch. It also needs estrogen hormone. So here's how we start doing the dance of creation, right? Is we say, yeah, we got all the genes necessary, and they're going to turn on over time and space, and that pathway is going to get regulated. And so you could think of this as being like, let's look at the estrogen receptor and watch its regulation. And it's not just the transcription of it, right? Could get regulated here, could get regulated here, and then here, post-translational regulation of the estrogen receptor is huge. If there's no estrogen, right, it's just going to repress its target genes like that. And that's so true. And so from a molecular biologist perspective, when you say, let's talk about the cell, they just say, yeah, well, let's talk about how the genes start getting expressed, and there's this potential for incredible amounts of interplay and feedback and adjustment like that. And that's what creates this process of creation, you know, that it's not just like, here's the genes, open them up, you know, boom, there it is, there's the organism. But it plays out over time, and yeah, the external environment can be a factor in regulating it, but that internal environment, the hormones, and even within the cell, the intracellular environment is massively important because the transcription factors get made, they're going to go in and do their job. But are they going to get, is it FOXO? Is it going to get to go in and do its job or not? And so that's going to be kind of a theme that'll drive us through this cell biology is, well, yeah, we're studying cell biologists from this molecular biology perspective. So the course is cell and molecular biology, and it's really dominated by molecular biologists watching cell biology take place. So we got to add that in order to get a feeling for how this could happen. You know, how could this be happening on our planet, right? All these new flowers that are about to come out, leaves and stuff like that. Oh man, it's pretty cool, right? Like that, okay? So we're looking at this, and it's, again, it's an intro bio concept, right? But in intro bio, you're just like breezing through these things. You know, here's a chapter, like that. So we're going to dive in some here. So we have transcriptional regulation. That's the first half of our course. And now we want to try to lace this into the cell biology chapters. So we'll be doing some of what's done in the RNA processing assignment, which is to use our knowledge of genes and put those in place into the nucleus and say, yeah, that's where transcription takes place, and also that's where RNA processing takes place. And there'll be specific examples of genes that are regulated in that RNA processing pathway. So we want to learn some about those, like that. And then protein synthesis, that chapter will focus on that, you know, and the potential for regulation. Well, expanding and talking about just, you know, how do ribosomes function and stuff like that, some, but our focus is going to be that regulation. And then cell membrane, yeah, same thing like that, you know, okay? So that's where we're going. And so gene expression has many steps to it. Each one has the potential to be regulated. If regulated, that step can be the rate-limiting step for gene expression, for gene action, for the way that that genotype, that gene affects that phenotype. That can be regulated at each of those steps. And there are specific examples. So we focus on the first step, and we're going to loop in some of the post-transcriptional regulation as we roll forward, like that, okay? And so that's just that figure, and I was going to write on here, you know, there's RNA processing, there's leaving the nucleus, there is translation, post-translational regulation. The biggest theme in chapter 10 is actually that the nucleus is, that nuclear membrane is a gatekeeper, that you have to have an ID to get through in either direction. You get carded at the nuclear membrane. And so that adds potential regulation. You like that? Yep. We've seen a couple examples of that already. We'll see in more detail, you know, what's keeping FOXO out of the nucleus, like that. It doesn't have its ID with it, so the bouncer at the nuclear membrane says, yeah, you just can't come in here. Can't come in, can't do your job. So we'll see examples of that. So here's the chapter. It's got really 3 parts to it, and we'll spend almost all the time on the nuclear envelope. That's the nuclear membrane and the traffic that goes in and out of the nucleus. Like that. Then there's going to be some about what are the chromosomes doing in the nucleus, just a little bit about that. And then within the nucleus, there's some organization. So in general, we've talked about how during mitosis, you can see the chromosomes. And during interphase, under a microscope, you can't see the chromosomes. And during interphase, the nucleus just looks gray, almost entirely gray. There is one structure that's seen called the nucleolus. We'll talk a little bit about that. But in general, it just looks like a really boring thing, you know? And so, you know, scientists were like, I don't know how much interesting stuff could happen in there. It doesn't look very interesting under a microscope. So what's going on here is scientists have come up with different ways to stain and tag proteins, like we talked some about, or stain and tag molecules, and be able to see more details. And so that's what these are. They're just examples of how, actually the nucleus is organized and has some organized locations in it, and we just kind of breeze through that, okay? And so here's how things get in and out of the nucleus. The nucleus is a double membrane, so this is a phospholipid bilayer, and it's a double membrane, and the traffic that we're going to talk about goes in and out of things called the nuclear pores. And so they are structures, and they're a great example of cell biology, because they're made of a whole bunch of different proteins, and that structure confers that function, you know? And so it's got a very interesting and complicated structure that serves as a gatekeeper, like that. And so that's what we want to talk about, and we're going to talk about getting in and out, okay? And so the first thing to say is your ID, you have to give to the bouncer at the nuclear pore to get into the nucleus. If your protein is, you have to have something called a nuclear localization signal, or an LSS, NLS. So who's got those? Every transcription factor has to have a nuclear localization signal. I'm just going to skip to an example of what the nuclear localization signal looks like, because I think it's up ahead a couple of slides. A few slides, maybe. Oh, there it is. So the nuclear localization signal is actually really short, and it's just a series of positively charged amino acids, lysine and arginine, in fact, and so it's very easy for a protein to have a nuclear localization signal if it needs it. So does FOXO have one? Yep. Do all those transcription factors have one? Yep. And it becomes, we wouldn't really call it a domain. You know, we talked about the DNA binding domain and the ligand binding domain. This is just a short stretch of amino acids. So it would just be a signal, and it could be anywhere really in the protein. Remember how we talked about with the heat shock transcription factor, we said that's always present in our cells all the time. Heat shock transcription factor is in our cells, and in the absence of stress, it's present as a monomer in the cytoplasm. And then when there's some stress, heat or other kinds of stress, it will start unfolding proteins, and it does change the folding of the heat shock transcription factor a little bit. And so far, what we said is that'll expose the trimerization domain. So there's actually a region that will bind the heat shock transcription factors together and make a trimer, and the individual DNA binding domains are weak at holding on to the heat shock enhancer, the heat shock response element, but when you have a three-handed climber, they won't fall off the climb, you know, like that, right? Okay, so at the same time that the trimerization domain gets exposed, so does a nuclear localization signal. So we can add to that, you know, the regulation of heat shock transcription factor has got a nice switch to it. It's in the cytoplasm as a monomer in unstressed settings, like every cell in our body and every cell and every eukaryotic cell on our planet right now. And it's stuck in the cytoplasm because its folding is covering this simple nuclear localization signal. And it will not be allowed in through the nuclear pore. So it's stuck out there, right? So that's an addition to what we already learned about regulation of the heat shock transcription factor. Then the heat stress changes its shape, and yeah, it trimerizes, but also really, essentially, it exposes the nuclear localization signal, and we're gonna meet a protein will bind to that and bring it in through the nuclear pore. So it's an escort. And that's the way the gate works all the time, is to come into the nucleus. You gotta come in through a nuclear pore, and you have to have a signal that will allow a protein to bind to you and help you come in, like chaperone you into the nucleus. And there's some proteins that help do that. There's a bunch of different proteins, and we're gonna learn the name of one of them. And I'll just put it here. I've got some slides for it too, but just to show you the name of the one. So getting into the nucleus involves getting a helper protein to bind and help bring you in. So there's the nuclear pore. And we could have, you know, the heat shock transcription factor trimer with its nuclear localization signal exposed. And there's going to be a protein that binds to it. And together, they come through the nuclear pore. And that's what happens every time. And there are several different categories of proteins that do that. Their job is just to bring them in, like that, right? And the only one we're gonna learn is, because it's just got a great name, is that protein is called important. So see, it's important and it's cute, right? So, you know, there's some graduate students here and there that are just cool people, you know, and you're like, well, I'm studying this protein and we call it important. And I'm like, thank you, that makes it easy to learn. It's for importing stuff, and it's fantastic, okay? And there are others, and the book lists a table of them, and you don't have to learn the others. Don't worry about the others. If we can get a handle on this one, great. And the only thing that's going to be a detail that we're going to add to this is we're going to see that, oh, the important comes in, it lets go of its cargo, because that's what people call it. What it's bringing in is called cargo. And then the important has to go back out through a nuclear pore. And it has to get back out by binding to a different category of protein that's going to carry it out. Because everything that leaves the nucleus also has to have a helper protein to give it permission to leave. And there's a table with a bunch of different names for these proteins, and we're just going to learn one. And these are legitimate proteins, and you're just like, okay, cool. Let's take exam 3 today. Because this one that we're going to learn is called exportin. Like that. Yep. And exportin is going to help. And again, you know, we'll use it. There are others that help, but it's going to help. It's going to help that important get back out. So the important is on a cycle, and that's just like, it's just cycling in and out. And when it's bound in the cytoplasm to cargo, it's allowed to bring it in, and then when it's bound to exportin, it's allowed to leave, and so we just have a steady amount of importin going in and out. And if you think about it, the same is true for the exportin. It gets out here, that's maybe a little bit wrong color. It gets out here, and then the exportin has to get back in, so it's carrying stuff out, lets go of it, and has to get back in through another nuclear pore. And so that's what these helper proteins are doing, is they're just bringing stuff in and bringing stuff out, and they themselves have to cycle. And we'll look briefly at the cycling process just for important, like that. Just a little foreshadowing, and that is that one of the things that these exporting molecules take out is they take out all the RNA. And we haven't talked about it before, but in our cells, it's rarely in books depicted this way, but RNA in our cells, it always has a bunch of different proteins bound to it. And it's those proteins, and we're not going to learn the names of any of those proteins, but just the reality is that we never are exporting RNA. We're exporting the proteins that are bound to it, and they're identified. They'll have these exporting tags on them, and they'll be carried out. And you'll see in our homework assignment, we'll say RNA processing happens inside the nucleus. Why inside the nucleus? Is because the proteins that are bound to the RNA won't let it get out until it's ready, until the RNA processing done, and it's like a mature RNA, and it's ready to leave the nucleus. So in our homework assignment, it's especially focused on that. Just let that RNA get out of the nucleus. Yep, like that. What we like as molecular biologists is we like to see this and say, oh man, I freaking love transcription factors so much. And here's a way they get regulated. Like heat-shock transcription factor, FOXO, not all of them, but some of them get regulated whether or not they can get into the nucleus. And you're just like, dang it, biology is cool, like that, right? And then when we're over here, we'll especially think about RNAs. RNAs need to get out of the nucleus, like that, talk about some of that. So let me scroll back and then roll through some of these slides. Like this, so I'll go back to here and talk through some of this stuff, okay? So here's an electron micrograph, scanning electron micrograph of the nucleus, and it shows those nuclear pores. And I mentioned at the start of the course that it was really in the 1950s when scientists started using electron microscope to look at biological cells, and that's when cell biology really began. Because you're like, look, what do you think those are? And they really didn't have the tools for a while, right? But they grind stuff up, they try to centrifuge, they try to isolate, what are these pores, what are they made of, and can we get like a little piece of a nuclear membrane in a test tube so we could study what's going on with that circle there, like that. So that's the really story of the big birth of cell biology, is they're just like, get busy, figure out what that is, and it's a nuclear pore, like that. And so drawing in our book about the nuclear pore, like that, that's our big focus in the nuclear membrane. There's other things to learn about the nuclear membrane. The nuclear membrane is actually connected to the endoplasmic reticulum. So as its structure and function goes, there's a really interesting relationship between those two, like that. So our book talks about that. So, our book is always talking on a bigger package of learning that we could possibly focus on in our class. So try to gather some of that as you go along. Try to connect it to your intro bio, because you'll remember, I remember that from intro bio. Depending on your instructor, who you had, they have sort of their favorite things they like to talk about, and they might have talked about the nucleus some, and you learn some about it. We don't have to learn how to draw that. We're not going to learn that at all. It is a visual thing. It's with a lot of detail to it. It's very big, and the one thing I want to point out on this figure is that these proteins here, we'll talk a little bit about, because they're part of the nucleus, and they're the nuclear lamins, or lamina. And that's those polypeptides that are like, the polymers are like sticks that are like lumber, that used to make the sphere that is the nucleus. And so our book wants to talk about some of them, and they're important in the overall shape of the nucleus, and we saw that these are the guys that get phosphorylated by CDK1 and Polo and Aurora, and they disassemble into monomers, and the nucleus breaks down into small pieces for lots of eukaryotic cells during M phase. So that's what we want to get out of this figure, is there's the nuclear membrane, it's a double membrane, and inside of it, it's being organized by those nuclear lamina, and the nuclear lamina also help place and space the nuclear pores along the nucleus, like that, okay? And so see, for example, our book just puts things on here, great, you know, but not our primary effort to try to connect our book with our class, okay? So here's this lamin protein, like that, showing how it polymerizes, and our book is explaining that, and where it fits in the nuclear membrane. And then cell biologists love to do this, where they're like, looks like the nuclear lamins are just a structural component. Just structure, nothing special about them like that. But it turns out that in some human inherited genetic situations, the mutations are in the nuclear lamin gene. So you're like, so, you know, if there's a mutation, you know, loss of function in nuclear lamins, what would the problem be? And the problem would be something with just the structure and organization of the nucleus. Maybe the nuclear pore function? I don't know. But here's an example the book talks about, and it's a very famous example because there's a really good chance you've heard of these children who have progeria, and they age like 10 times faster than normal. So this is a young child who's maybe 5 or 6 years old, and they already look like an old person. And in every way, their body is just aging faster than other humans. And what's their problem? Well, it's inherited genetic disorder, and scientists have mapped where the mutation is, and it's in the nuclear lamin gene, right? Yeah, right. So these kids are born, they're healthy, they have good bodies, they just age super fast. And then that, all the things with aging pile up in them, right? Heart disease and so forth like that, you know, really fast. And the problem isn't that, you know? And so this is classic cell biology, where you just put your hands on your hip and you're just like, what do you think about that, you know? Cell biology is super cool, because who knows what's going on there? And scientists have learned a lot about it because it's been known for several years. And there isn't a good summary explanation yet. Like, what is wrong? You know, what's going on? Like that. The mutation is also something that we want to just focus on a little bit. So, because in our RNA homework assignment, we're going to talk a little bit about gene splicing and exons and introns. And these children have a mutation that's just in one specific part of the coding region for the nuclear lamin gene, like that. And so they want to zoom in on that and talk a little bit about that in this chapter. And again, the book talks about this RNA processing in chapter 6. The issue is an issue of RNA processing of the gene. It makes a different protein because it puts together different parts, or not all the parts of the RNA. So also a good mutation for us, because it's also going to be talked about and as an example in that RNA processing assignment. It's a different kind of mutation than the kinds we talked about before, and it just happens to be a good example for that reason as well, okay? And so then, you know, just more, the book really wants to talk a lot about the structure and start naming these parts, but, you know, just we don't need to go there, right? It's a gatekeeper, and you need to get in and out of it, and that's what we want to focus on, okay, like that. So here's just an example of maybe a better view of what a nuclear localization signal looks like. It's just positively charged amino acids all clustered together, like that. Very simple thing. What is that? It's a binding site for proteins like important. Yep, that's where it would bind. That's the nuclear localization signal that the important needs to be able to see and bind directly to, like that. And so here's just, starting to introduce this, that we need a transport protein, like important, to bring stuff in. We need an export protein to take stuff out. And we have important, bringing stuff in, and export and bringing stuff out. And the book's showing that, not everything, there are some small molecules that can get out of the nuclear pores, but for most of the stuff that we care about, you gotta have these nuclear localization signals or the opposite to get out, like that. So there's that table again. And then here's just the pathway for important. So there's that important protein binding to a cargo protein like heat shock transcription factor or anything else. So a list of what needs to get into the nucleus is really long. All transcription factors, histones, condensins, cohesions, all the proteins of the chromatin need to get in. DNA polymerase, DNA repair enzymes, MCM, all have to have a nuclear localization signal or they will not get in. I don't know of any proteins that get in without that cargo carrying important or another member of that group. Yeah, so lots and lots of proteins have this nuclear localization signal. We'll focus just on the transport protein, the helper protein important. Here it is binding and bringing it in, and then it lets go and goes out again. And I just want to show you this, that to go out again as part of this cycle, it actually has to hydrolyze, some GTP has to be hydrolyzed. GTP is just like ATP, we're gonna burn it, it's gonna do some work, like that. And so just to pause on that for a minute, so we're used to thinking about ATP or GTP, same type of thing, as, oh, that's important, you need ATP to do physical work and move around like that, And you need ATP to synthesize, to build biological molecules. And this is a really nice example to see that what's going on here is there is a kind of cellular work that's really focused on information pathways. And this is a kind of information pathway because it's like this information of what do you need to get in. And we want the pathway to go in a direction like that. It just keeps cycling. And the way to think about burning it is like thinking about the work that it does to turn a light switch. In a sense, that work is more kind of information work. We're not moving around the room. We're not building DNA and other molecules. We're using the energy to make a switch and make the switch so that the cycle flows in that direction. And just having one switch is enough to drive the whole cycle in that direction. And so for us, important, we want to know that. And we also want to know that it just goes to go in and out and in and out. And one place on its cycle, it requires the burning of GTP to put an energy switch into it. So the flow is that direction. And it's true for all the other guys that are bringing stuff in, is they need to have an energy switch along the way, like that. And we'll talk about that once or twice more because it's interesting to see when people talk about, wow, life is complicated. There's lots of information flow in it. The information flow is chemically connected to energy use. And the energy is used to create a kind of information path, like that. So that's what's going on here, is the importance doing its job. To do its job, how does it even know which direction to go? Should I come in or out? And the answer is, well, you're mostly out. You should come in. And when you come in, we're going to push you out, like that. And you're pushed out, so come on in, push out, like that. Or like a kid, with a merry-go-round, and the kid's just standing in one place, pushing around the merry-go-round. They're off the merry-go-round, and they're like, we're just pushing the merry-go-round. And the merry-go around, and come back, push it again, like that, yeah. So that's, one important thing about important is that the cycle requires energy. GTP hydrolysis requires that energy intermediate to get that job done, okay? And that's it. These details here, don't worry about them, right? Importance in the cytoplasm, it binds to a cargo that's got a nuclear localization signal, that's enough to get it in, then it lets go of it and burns GTP to get back out. Yep?
Is the cargo protein just any protein that needs
Yeah.
or is it another?
Yeah, yeah, cargo is just like a, you know, a term, loose term for whatever it's gonna bring in, and the only thing it'll bring in is a protein that has a nuclear localization signal. Yep.
Does it burn the energy before, or the GTP before it binds to exportin, or like?
I'm not even sure. That's a good question, and I'm not sure, so I don't wanna speculate.
Yeah, okay.
Yeah. But it would be in that, you know, it would be in that, right, it may be like right in that. It's definitely on the, you know, in the nucleus where it's gonna need to bind exportin to get out. And whether it's, you know, physically coupled to that or before or after that, I just don't know for sure. But somebody does, you know, like that, you know, AI does. You guys should quit college, just stay home and Google. Yep, so we wanna be able to just draw that. Not this complicated, but like a nuclear membrane with a nuclear pore here and here, important, the cargo, what is an example of cargo, you know, use, you know, like on the exam, list, you know, eight examples of cargo for important, you know, and you just list anything that has to get in. It's gotta have a nuclear localization signal, like that, you know? So we can ask that question a couple different ways, but it's just that. We just wanna focus in on that and not, you know, sweat the details. Like, for example, that it's this RAN protein that's actually the one that's got the GTP. So it's actually a GTP protein. Don't worry about it like that. Just that it burns GTP to make the cycle go. Yep. All right, super duper. And so here's this table. Here's importance, important, transportant, important 7, exportant, exportant T, exportant 5. You know, just don't go there, right? Even if, you know, yeah, just don't go there. Important, export, fantastic, like that, right? Here's exportant doing its job, like that. Also has to burn GTP to do the cycle, like that. Get super, lots of stuff going on. Just draw the cycle, 2 pores, export, need some cargo. The cargo in this case could be newly synthesized proteins. They have to get out, right? So, right, so when we, like, remember back when we said, oh, MyoD is a gene, and it's got to get expressed. So you got to make MyoD, and it's got to get out of the nucleus. No, not the MyoD, RNA. The RNA has to get out of the nucleus, then the protein's got to get back in. So there's a lot of this cycling as part of gene expression, so there's potential for regulation. Regulation of letting the RNA out, letting the regulation of the protein, the newly synthesized protein back in. Great. And this is the book showing examples of the regulation of getting in. And they happen to use this transcription factor that we did not emphasize. So back in chapter 16, we learned about some receptors and signal transduction pathways. At the end of the chapter, we learned about some individual receptors like Notch and the Wnt signaling pathway and SMADs and non-tyrosine receptor kinases. And it turns out there is one that is that uses the TNF ligand and activates this transcriptor NF-kappa B, and it's just chosen here. We don't need to start from scratch and say, oh yes, I gotta go back and learn all about NF-kappa B. No, we can just say, I wanna focus on FOXO or heat shock transcription factor. The easiest one for us to use in these kind of, you know, explanations, just use heat shock transcription factor. Because change its folding, it's gonna trimerize, and it's also exposing its nuclear localization signal, yay. Can't get in, gets in, fantastic switch. You know, just a really satisfying switch. Or FOXO, but, you know, could be any of the ones that we've learned about, okay? Like that. So, so see, the book is diving in. And again, the book dives in with details, we're set. We've got a good examples, good explanation, good stories, good examples of the important, good examples of the export, like that. So I just put this up, right, because FOXO, right, that's our first thing, our first, you know, figure from the first day of class. And there's the nuclear pore, right? It's like, oh, you gotta get through there, I bet, huh? And what's going on when 1433 binds? What do you think? Well, it's blocking the nuclear localization signal. That's what 1433 does. Blocks nuclear localization signals like that. Literally just covers them up. And to bind FOXO, it just needs to have to be phosphorylated. And we mentioned before the second exam, we were talking about the cell cycle. And there's that CDC25 phosphatase that's gotta remove inhibitory phosphates from CDK1. I just mentioned briefly that it turns out CDK1 is primarily localized in the nucleus, and CDC25 gets made out here, like other proteins do, and if it gets phosphorylated, it can't get in. Because CDC25 phosphatase must have a nuclear localization to get in to phosphorylate its target, or to remove the phosphatase from its target. And 1433 does that. So as part of this, on our examples, we actually want to talk a little bit more about 1433. Because I mentioned, whoa, those are different stories, right? FOXO, transcription factor, CDC25 phosphatase. So let's just talk a little bit about these 1433. So it's a gene family, like that. So several paralogs, and that's because this regulation is really effective. We got a protein that needs to get into the nucleus to do its job. How about if we just sequester it in the cytoplasm? And that's, I think that's the only thing 1433 does, is it can bind to proteins and have them stay in the cytoplasm. And you can just think like ancestrally, you know, it just was, that's a good thing, you know, and maybe it'd be helpful to have a gene family, you know, with some slight different features to it, but they basically are involved in that, controlling that, like that, okay? And so here it says regulate cell cycle progression by interacting with and modulating the activity of CDC25 phosphatase. You know, blah, blah, blah. So we learned that for our second exam, and we're learning more stuff for 1433 for our third exam, because we're talking about the nucleus, and 1433 is a player in the cell biology of the nucleus because it's blocking nuclear localization signals in a switching way, right? Okay? So this would be an example of gene expression. FOXO gene expression can be regulated even after the protein gets made, because the protein might not be able to do its job. And it can't do its job if it gets phosphorylated, and 1433 interacts with it, binds to it. So then we really get really interested in trying to think about, like, well, what is a list of all the proteins that would get bound by 1433? Like, well, I'd like to know all of them, because it's a lot. So remember, up to now, when we start talking, oh, it's a lot that it's regulating, those would be examples of target genes, and that transcription vector is regulating a bunch of target genes. So this is not that. It's a different kind of example. It's a cell biology example, where a family of proteins is regulating a bunch of, and we won't call them targets, because that can get confusing with target genes. It's regulating a bunch of proteins. And it would be cool if we could find all of them, you know, study them all. So scientists are working on that, like trying to really understand that, right? So CDC25, that's one of them. It's regulating that. FOXO, regulating that, you know, that's a good 2-point question. You know, 1433 regulates FOXO, NCC. 1433 binding can inhibit CDC25 activity by preventing its translocation to the nucleus, where it would activate CDKs. So a little bit more from chapter 2. You know, you don't have to go back and start over learning chapter 2 material, but we're taking examples with us as we move forward, like that, okay? Yep, and so just to say this, you know, ADB, yes, CDK1 is present in the nucleus, participating particularly during mitosis, you know, so that's where you're gonna find it. So just, you know, a nice cell biology example of, we're talking about the cell cycle regulation, and it's a G2 to M transition, and CDK1 is the action protein, and not too surprising to find it in the nucleus. It's got to phosphorylate all those histones and cohesions and cadensins and nuclear lamins. You know, got a lot of targets in there. So that's where it is. And you got to remove those inhibitory phosphates. But can CDC25 get to it? Not if it's bound by 1433. And 1433 loves to bind it when CDC25 is phosphorylated. And it gets phosphorylated by check 1 when there's DNA damage. So just to kind of flesh that out a little bit, this gets way too busy, right? You know, there's more to that story. You know, our course is still, it's not intro bio, but it's still an introduction to molecular and cellular biology, right? And so everything we're saying is correct. It's not an oversimplification, but it is the introductory view of the role of CDC 25 and the regulation of it here. Because here it is, it's just saying there's CDC 25, we've got a lot of different stories we can talk about. Here's the 1433 story and sequestering that in the cytoplasm. How's it do that? CDC 25 must have a nuclear localization signal. It's got to get into the nucleus, that's where its target is, CDK1 is. CDK1 itself must have a nuclear localization signal. You just have to have it, you're going to get into the nucleus. And can't get in if it's bound to 1433. Okay? Same is true, exactly same story for FOXO. It needs to get into the nucleus to do its job, can't get in if it's bound to 1433. All right? And so here's just a paper that is talking about 1433 and the idea of, my gosh, that family has evolved to do this same story in a lot of different situations, a lot of different proteins, from FOXO to CPC25 to lots of others. And you'll see there's a little bit of information here of this family. You know, it's still a family, it's still like they're screwdrivers, still do the same kind of thing, but families can evolve to have slightly different but very diverse functions. And that's the case for this 1433. It's just so many different things it does, like basically doing stuff like that, right? So here's just an abstract maybe from that same thing. It says 1433 proteins are a family of structurally similar phospho-binding proteins that regulate essentially every major cell function. And you're like, whoops, you know, like that. Decades of research, blah, blah, remarkable network, interacting proteins like that, you know, so just, my gosh, it's just massively significant in that story of getting through the nuclear pore, right? That's what we're talking about, like that. And so here they use this term, it's the first time we use it, we want to see this, and they call this the 14.3 interactome. And what that means is, let's try to find all the proteins that 1433 family members bind to, that they interact with. And the word interactome is derived from, right, you know, the word genome, and genome is all the DNA needed to build, a species. So we got the human genome, so forth, and so this is, they're interested in something they like to say, let's talk about all the proteins that 1433 family members could interact with, and if it's all of them, we'll call it the 1433 interactome, like that. Because again, it's just a massive number of examples and stories of all kinds of different things. And, many, maybe all of them are based on, you going to get in through the nuclear pore or not? Like that, right? Okay? And so it's just talking about how can we study that, right? So you need different kinds of tools to study something like that, of all that protein interaction. That would be an example of post-translational regulation. FOXO is regulated after it gets synthesized, like that. And this is an example of that, 1433 is involved in regulating entry into the nucleus of proteins, cargo, and those proteins are all regulated in that way. So the pathway of gene expression for FOXO includes very, very late regulation at the level of, can you get to the location you need to do your job? And if you can't, we don't even have that function. The FOXO function isn't even occurring, like that. Yep?
What's going to drive 1433 to bind to something to not let it in the nucleus?
Yeah, so that's just, and we haven't really talked about this, but molecules interact through non-covalent binding. So each molecule is composed of covalent bonds, and whatever the two molecules are, it's through non-covalent interactions. And we'll just use a simple example to describe that general concept of these non-covalent binding. And I should say our book talks about that binding a little bit in chapter 2. Chapter 2 is about chemistry. And so when we talk about that binding, we're talking about binding coefficients from chemistry, like the association and dissociation constant, Km and stuff like that from chemistry. You learned about it. But it's true for all these interactions. So just for one, well, kind of describe this one. So the insulin protein it's a small protein, but if a cell was as big as the Boston metropolitan area, insulin would still be the size of a truck, like that. Let's say a Cybertruck, just because it's a pretty big truck, and we can get political or whatever, pretty big truck. And the only thing that matters on insulin really is the grill, because the grill has a certain shape and chemistry to it, and that's actually the only part that binds the insulin receptor. And the insulin receptor has a binding domain, it's just a pocket. We can park the front end of that truck there. And what does that mean? What does it look like? And the answer is the insulin receptor is made up of amino acids, and the binding pocket, the ligand binding domain for the insulin, is just a surface of amino acids with the side chains that have some chemistry. Generally, a binding pocket like this and the grill of the insulin protein are made up of a lot of hydrophobic amino acid side chains. So they tend to stick together because they're in an aqueous environment, a hydrophilic environment. And so one reason that the insulin protein is pretty big is because the rest of the truck is pretty hydrophilic. The amino acids covering the rest of the truck, pretty hydrophilic, pretty happy to be dissolved in blood, but that grill is not happy because it's got a lot of hydrophobic amino acids. And those also have, it's a certain shape that can fit into the binding region like that. And so that would be an example of like what would be like for 1433 to bind to cargo, is like that. There's a really strong likelihood that both those would have some regions of hydrophobic interaction, like that. Okay, so then the next thing to say is that the insulin receptor have what is called both specificity and affinity. And it has a pretty high affinity. We don't want insulin binding to other receptors. We want it to bind it with specificity and high affinity to the insulin receptor. And the way that happens a lot is this. So, overall the pocket and the truck grill are the same kind of shape, and they're both hydrophobic, so we just kind of park it there. But then, really typically, there'll be some other amino acids that will increase the affinity and the specificity. And a good example of that is on the grill, right there, there's one positively charged amino acid side chain. In the pocket, right there, there's a matching negatively charged amino acid. So yeah, hydrophobic, let's bump into each other. Yeah, it's kind of a good place to get stuck because of the water environment. And whoa, those matched up. And as they match up, it's positive binds to negative. And when that's a salt, that will dissolve really fast because the water loves to dance with the positive and negative charges of the chlorine and sodium, like that for salt, like that. But when you have the truck grill bump and park in that insulin receptor binding domain, now, because it's hydrophobic region, as it binds, as it parks, water molecules get excluded. And now, that positive and negative make a kind of salt bridge that's very stable. So just with one thing like that, you can have very high affinity and high specificity because they're perfectly next to each other. One in the receptor and the other in the ligand, the insulin, like that. And then there could be other examples of that too. There are some amino acids that have hydrophobic side chains. There could be a couple of those that also match up. Those also get greatly stabilized when they end up in a hydrophobic environment. So water's going to get pushed out. And so that's really, it's true that insulin binds to insulin receptor, but the details of its binding are, I don't know if it has a positive or negative charge, but it's very common where proteins are going to interact like that, and they have a surface of amino acid side chains, and the amino acid side chains are complementary like that. And out of that, we get high specificity, high affinity. So the same would be true here. Okay? So, you know, what do you think about these nuclear localization signals? Well, one is, we mentioned that they have a lot of, just a stretch of positive charges. So I imagine, you know, that the binding site on important that helps it recognize nuclear localization signal is a cluster of negatively charged amino acids, right there. So that's probably part of the story, is that the nuclear localization signal is there, and then it's probably surrounded by some hydrophobic amino acids, and you make a kind of binding pocket, just very simple, not specific, because you have nice specificity between the important and its negatively charged amino acid side chain sticking to the positively charged nuclear localization signal. So it would be stuff like that, and again, I don't know the details of the binding, but yeah, and you get this association, dissociation constant, right? It's going to bind with a certain average and then let go just spontaneously like that. So I have a certain affinity like that. Yep. Yeah. And so 1433, and what does it interact with? Okay, and let's see how we're doing. We have to stop. So that's a good place to stop. We'll keep talking about this on Thursday. I will get the